HHT. And firstly, I briefly want to go over the history of HHT just to put things into context. So it was first discovered and characterized in 1864 by several English physicians, Babington, Sutton, and Legge. And they really identified the hallmark characteristics of HHT, the nosebleeds. And they also noted that it was a hereditary disease. In the late 1800s and early 1900s, three doctors, Rendu, Osler, and Weber, continued to further characterize HHT, and they really identify things like telangiectasias and arteriovenous malformations, and they really just further characterize the disease. So between 1907 and 94, not to say that there wasn't many things done, there just weren't many historically relevant things done in HHT. There were many doctors between 1907 and 1994 that contributed to the disease and treated it. But it wasn't until the early 1990s, the advent of modern technology and science that allowed us to identify and discover the genes that actually cause HHT. So we have here in 94, 96, and 2004, the endothelium gene, the ALK1 gene, and the SMAD4 gene. So to understand the biology of HHT, we have to start at the basics. Cells, DNA, and proteins, what they are, what do they do, and how do they relate to HHT? So this is a nice graphical image of a cell. And cells are defined as the basic building blocks of all living things. The human body is made up of trillions of cells, and there are roughly 200 different types of cells. And just to give you an example, here we have an array of different cells. We have motor neurons, white blood cells, we have reproductive cells, so on and so forth. Everyone is very familiar with the white blood cells. They kind of defend against infection to an extent. So they all have a specialized function or job to do in our body. So this schematic diagram is a hierarchical organization in the body. So we start at the very upper. Let me see if I can get a pointer on the screen. There we go. So we have atoms, and atoms are kind of organized into molecules. And then molecules are organized into cells, these single unicellular things. And they organize or rearrange themselves into tissues. And then these tissues are further organized to form organs. And organs have a function, a purpose in our body. For example, the lung is made up of many different types of cells that work together to make tissues. And then these tissues then form the organ. And a great analogy for an organ is a hospital. So a hospital is composed of many different people, doctors, nurses, custodial services, and culinary services. And they all kind of represent different cells that may exist in an organ. And they all have a particular job or function to ensure that the hospital or organ can work properly. And cells on average are 25 micrometers. So there's 10,000 times smaller than a centimeter. And just to put that into perspective on this size scale diagram here, we have cells over here between 10 micrometers and 100 micrometers. For human cells, we're roughly at 25 micrometers. And we have an ant over here at 1 centimeter. So we could approximately say that a cell is 10,000 times smaller than an ant. So our next topic is cells and DNA. And this is a very nice image of a DNA. And I'm assuming most people are familiar with this image, the famous double helix shape of DNA. But what really is DNA? So it's found in almost every cell in our body. And I like to define it as a library of information or instructions. And this information or instructions is stored in the form of genes. So a good analogy for DNA is that DNA is like a bookshelf. It is a structure that holds information. What information exactly? Well, information to form life. Everything that a cell needs to function properly is in the DNA. And each gene is kind of like a book on that bookshelf. It's a source of information or instruction. So I like to think of genes like the blueprint for a home. So the blueprint is information. It's instructions to build the fundamental units of a house. So just like the blueprints hold information to produce the structure of a home, the genes hold information to produce the fundamental units of a cell, the protein. And these proteins allow the cells to exist and function. So what exactly are proteins? In our current generation, it is a very hot topic, especially for those involved in fitness. But we're going to dive a little bit deeper and understand what protein does in our cells and our body. So what do they do in cells? They're responsible for most of the work in a cell. They make up the structure of a cell, and they ensure that cells function properly. And each protein is made up of amino acids. There are millions of different types of proteins, and they carry out thousands of different jobs. And they all have a job to make sure their cell works properly. So, for example, if a car is a cell, then the proteins would be all the different parts or components that allow it to function, like the wheels, the engine, and the mirrors. So just to summarize what we've learned so far, there are millions of cells in the body. Most of them have DNA. And DNA is a way to store information or instructions in the form of genes, which I like to think of as blueprints. And these blueprints or instructions carry information to make proteins. And these proteins are necessary for the cells to work properly. So what happens if a gene is mutated? And what does mutated mean? So if genes are like blueprints or instructions to build things, the proteins, what happens when these instructions or blueprints are incomplete? So in this cartoon here on the side, let's assume that the instructions here were to build a wardrobe. And you can see that the wife is complaining, it's very good, but we bought a wardrobe. So let's assume that this guy is competent and that the instructions were actually incomplete. So the instructions were unable to allow him to build a properly functioning wardrobe. So we're left with this half-looking wardrobe-like thing that doesn't really have a function or a purpose. So what happens when the genes in our cells are mutated or a gene is mutated? So let's mutate these genes. And the lightning bolt will signify a mutation throughout the presentation. So when we mutate a gene, the instructions are going to be incomplete. They're kind of unreadable. And these instructions make proteins. So then if the instructions are incomplete or unreadable, then we're either going to get broken proteins, just like that wardrobe, or we're not going to get enough protein being made. And this could have negative consequences on the cell. The cell is not going to work properly because, like I said earlier, these proteins have very fundamental, essential roles in the cells. So here's another good analogy. Again, if a gene is an instruction, this particular instruction reads, replace the oil in your car when it gets old. Now let's mutate this gene. So now the word oil is gone. Now we have replace the blank in your car when it gets old. We don't know what to replace now when it gets old. So what's going to happen? The car is most likely going to break down. And this is a kind of an analogy for what happens in cells when a gene is mutated. So we've seen throughout the talks yesterday and today, and we know that these three genes are the very prominent HHT genes, endoglin, alpha-1, and SMAD4. And there are at least 3 genes involved in HHT. These are the most well characterized, and the majority of people will have these ones. There are other mutations that are less common and less understood, but for simplicity, we'll focus on the main ones. So just to recap, genes are like blueprints, and these blueprints make proteins. And in HHT, if we mutate these genes, then their protein is not going to work properly, or there's not going to be enough of it. And then the cell is not going to work properly. And I want to be clear that HHT patients don't get all 3 of these gene mutations at the same time. You're either going to get an endoglin mutation or an ALK1 or a SMAD4. But really, how do these gene mutations cause HHT? So if we refer back to our summary diagram, we have cells that have DNA, that have genes that make protein. And when we mutate these genes, there's not going to be enough protein, or the protein is going to be broken and the cell's not going to work properly. But the question is, in which cells are being affected by these gene mutations? Like I said, there's trillions of cells and there's 200 different types of them. So which cells are being affected by the mutations? So let's kind of work backwards. What is HHT? Well, again, we're very familiar because we had a lot of great talks this weekend. And we know that HHT is a blood vessel disorder. And it leads to bleeding and these blood vessel abnormalities, such as the telangiectasias and arteriovenous malformations. So that must mean then that these gene mutations disrupt the normal function of cells that make up blood vessels. And that's in fact the case. And these cells are called endothelial cells. And that brings us into our next topic. So what exactly are endothelial cells? What role do they play in our blood vessels? So this is a very nice graphic image of a blood vessel, and it's a cross-section for us. So we can see its different layers. So there's an outer layer, there's a middle layer, and then there's this inner pink layer. And the stuff moving through the blood vessels, of course, blood. These are red blood cells. So the endothelial cells, endo meaning inside, actually make up the innermost layer of the blood vessel. So it's the pink layer. So here's another perspective. We're actually inside the blood vessel right now. And you can see the red blood cells flying past us. So this inner layer that we're looking at, this is the endothelial cell layer. These cells join together very tightly to form a layer in the inside of the blood vessel. Another good analogy of the inside of blood vessel is like a brick tunnel. So if this tunnel is a blood vessel, then each of those bricks represents an endothelial cell. They join very tightly together to line the inside of the blood vessel. And here's another good picture. These cross-sectional pictures are very informative. So you could see that this arrow is pointing to the endothelium, or the endothelial layer. Each one of these guys here is a cell. So they join together side by side in what we call a monolayer. Side by side they join to make up the inner lining of the blood vessel. So that's great and all. But what do they actually do? So they're very important for the blood vessel integrity or structure. So to make the vessel leaky or not. And they're very important in blood vessel formation, or a process that we learned from Dr. Krings, called angiogenesis. So in HHT, the HHT genes, these genes make proteins that are very important for the normal endothelial cell function. So when we have a genetic mutation in either one of these genes, and there's not enough protein being made, or the protein's broken, the endothelial cells are not going to be working properly. So to summarize what we have, we have genes which are instructions. They make proteins, and in the case of endothelial cells, they allow, when they're functioning normally, they allow for a normal vasculature network. So we have red blood vessels, which are arteries. We have blue ones, which are veins. And they're normally connected by these smaller capillary networks, the intervening capillary beds. Now in HHT, when these genes are mutated and the proteins are broken or there's not enough of them, the cells aren't going to work properly, and we could get an arteriovenous malformation forming. So you could see that we lost some of the intervening capillary beds, and we're left with this messy, disorganized, direct connection between an artery and a vein. And I'll go back for comparison. So we see nice capillary beds intervening, and now we have an AVM. So we lose these capillaries. So, but why is it bad if we lose capillary beds? This brings up our next topic, capillaries and HHT. What do capillaries do in the body? Well, they infiltrate and disperse throughout the various organs and tissues in our body. They're a vast network, like roads throughout a city. And they supply our body with blood, oxygen, nutrients, immune cells for healing in the case of if you have a wound or infection. And they can also clear waste and infection. So on the left here, we have the blood vessel network of a lung. And it is in fact in the shape of a lung. And this really underscores how well blood vessels infiltrate every space of an organ. And the smaller ones are the capillaries. And so I like to think of capillaries as a road network. All the capillaries in this lung act as a road to deliver oxygen, nutrients to all the cells in the lung, just like the roads allow us to transport all sorts of things throughout a community. And how do capillaries do this? How do they grow into all the nooks and crannies of an organ to ensure it gets blood, oxygen, and nutrients? Well, this is through a process called angiogenesis, and we learned about it yesterday and a little bit today. And to break the word down, angio is referring to blood vessel, and genesis, it kind of means new. It could also mean birth or growth. So this kind of means blood vessel growth. And I have a nice video that really underscores this process. So this video is displaying the process of angiogenesis. This is a blood vessel growing from a pre-existing blood vessel. And in the context of this video, at the end here, we see that this is a wound that it is growing into. So this blood vessel is actually growing into this wound or cut to supply it with clotting factors, immune cells, and many other things. So blood vessels and capillaries are used like a highway to deliver necessary things to various places in the body. And this mechanism of growth, again, is called angiogenesis. So a good analogy is blood vessels are kind of like tree branches. The tree branches grow toward light to supply the tree with nourishment from the sun. Blood vessels will grow and infiltrate areas of the body to supply it with things that it needs. So this mechanism, known as angiogenesis, combined with the dysfunctional endothelial cells due to the genetic mutations, can cause, for an example, an AVM to form. But how does everything connect? How do mutations, endothelial cells, and angiogenesis all lead to HHT and the AVMs and these manifestations that we see arise in the disease? So we're going to discuss some mechanisms or processes that science has shed light on to discover how all these things connect together to cause the disease characteristics. And it starts with mice, of course. I believe Dr. Fawnin touched on mice earlier and their importance in researching HHT. So the discovery of how all these things connect together to cause HHT was really identified in mice. Scientific advancements in technology has given us great opportunities to study HHT. So we can, in fact, give lab mice HHT. We give them the genetic mutation, whether it's endoglin or ALK1. Doesn't matter. It depends on what we want to study. But what's interesting is that when you give mice these genetic mutations, they actually develop some of the physical effects of the disease. They'll get like fingernail bleeds and blood vessel issues, but they don't readily get full-fledged AVMs. So there must be something else that is contributing to the formation of AVMs and HHT. So scientists were very smart. They took an HHT mouse and they actually gave it a wound. They made an incision on its skin. And they found that when you do this to a mouse that has the genetic mutation, an HHT mouse, it has a higher chance of developing an AVM. So this led scientists to conclude that you need angiogenesis and the genetic mutation to get an AVM, or at least a chance of getting an AVM. And just to recap, AVM is the process of blood vessel growth from a pre-existing blood vessel. So they found that you need this process to be occurring, coupled with the genetic mutation that disrupts endothelial cell function to potentially make an AVM. So these blood vessels are made of endothelial cells, as we learned earlier. So in HHT, we know these cells aren't working properly. So during this process, there's a higher chance that it will cause an AVM, like so. So We also learned something else very interesting from these HHT mice. We found that, sorry, I say we, I'm referring to other scientists that have been, that have put in great effort to discover this, not me specifically. So scientists have discovered that a protein called vascular endothelial growth factor, if you actually induce that or give it to an HHT mouse, it could also lead to the development of an arteriovenous malformation. So it led scientists to further conclude that all you really need to do is trigger angiogenesis or blood vessel growth to get an AVM and HHT. And we learned earlier that this protein, vascular endothelial growth factor, is a potent stimulator of angiogenesis or blood vessel growth. And this new knowledge, this discovery led to the advent of anti-VEGF or anti-angiogenesis therapies. Dr. Fondon touched on a lot of these, and she did mention Avastin or bevacizumab. This has shown to be very helpful in reducing nosebleeds in HHT patients. And there are other drugs that have anti-angiogenic or anti-VEGF effects, and there's many more in the pipeline. So it's quite exciting that scientific advancements have, opened up the door to these new treatments. It's exciting how science can potentially directly help patients. So I want to do a final summary of everything we learned. I really hope that you have a better understanding of what's occurring in HHT at the microscopic level, at the cellular level. So if I refer back to earlier, we have our 3 gene mutations, and these genes are blueprints or instructions to make proteins. When you mutate these genes, there's either not enough proteins or the proteins are broken. And thus, at least in the context of HHT, the endothelial cells will not be able to work properly. So because of the mouse models, we found that if you couple this with an angiogenic trigger, so something that stimulates angiogenesis. So we learned about VEGF and we learned about wounding, 2 things that stimulate angiogenesis. There are other things, of course, but if you couple the gene mutation and the endothelial cell dysfunction with this angiogenic trigger, it leads to a high chance of developing an arteriovenous malformation or telangiectasia. So there's still a lot of things that we don't know for certain. So for example, why do affected family members have such different symptoms and severity? So for example, if a mother with HHT has a child who also has HHT, why is there sometimes a difference in symptoms or severity between the parent and child? Sometimes a mother who is mild may have a child who is severe or vice versa. So this is something that is somewhat unknown to us. Additionally, why do these AVMs or telangiectasias form in certain places or organs and not everywhere? Now, we have somewhat of an idea of why this happens, but it's still not 100% clear. So there's certainly a great deal that we don't know about HHT, and it is paramount for scientists to continue their investigation. So thank you, everyone, for listening. I greatly appreciate it, and I'm more than happy to answer any questions.
Okay. Thank you, Mr. Cannavicci, for that amazing presentation, which might hopefully inspire some of the younger audience to pursue a research career involving HHT. But so on to our questions. First question will be, do our bodies have mechanisms to compensate for these genetic mutations? If so, what are they? And also, she said, thank you for the great talk, very informative and clear.
Excellent. A very excellent question, and certainly a very challenging question to answer. The reality is that the body is always trying to compensate for faults or problems. And unfortunately, in HHT, because these genes, at least the proteins that these genes make, are very important for the endothelial cells to function, there isn't any other mechanism that can truly compensate for the loss or of that protein or the loss of function of that protein. So, I mean, there's lots of things happening in the body that try to compensate for it. But if those things were effective, then maybe we wouldn't have this disease at all. But to speak more on that, there are many other factors that are still being discovered. There's many other mechanisms in the body. It's very complicated, and it's possible in the future that we could discover exact mechanisms or pathways that are directly trying to alleviate this genetic mutation.
Okay, great. Thank you. The next question will be, do the different genetic mutations lead to different locations for AVMs?
Also a very good question. I'm certain the clinicians that we've had during the talk today would be better suited to answer that question, but I am aware that genetic mutations can lead to I mean, what I mean to say is that it is possible that there's a higher chance of developing certain AVMs per certain genetic mutations. But the reality is, and I believe that there's significant overlap. So like for pulmonary AVMs or brain AVMs, whether you have endoglin or ALK1, it is possible for you to develop either or with both genetic mutations.
Okay, great. Thank you. Another question is, what is your research focused on?
Great question. And I did consider talking on my research, but it certainly would have went a lot longer and possibly would have been a little bit more complicated. But I study factors in the body called microRNAs. So like DNA, like proteins, these are present in all our cells. These microRNAs are present in all our cells and in our body, and they actually can control how much protein is being made. So what I have been doing in my research previously, pre-COVID, I was recruiting patients at St. Michael's Hospital and growing cells from their blood and then measuring their microRNAs, so the microRNAs in the cells. And then I was comparing the levels of these microRNAs in HHT patients to people without HHT, or controls as we call them, and looking for differences. And once differences are detected, to further understand, okay, we have a microRNA that is, like hypothetically speaking, we have a microRNA that is higher in HHT patients compared to people without HHT. Why is this? Is it contributing to the disease? Is it a result of the disease? This research is ongoing. So unfortunately, I don't have any concluding remarks on it.
Okay, thank you. Another question, what kind of injuries would cause PAVMs or CAVMs? I understand how it may create telangiectasias, but not the type of injury or trauma that would cause the larger organ AVMs.
So again, a clinician would probably be better suited to answer this, but one comment I can make is that it doesn't only have to be injury to potentially stimulate the growth of an AVM. There are other processes in the body and other things happening that could stimulate angiogenesis to increase the chance of an AVM forming. But during our open discussion at, I believe it's at 1:40 or after this talk actually, that's a good question to ask.
Okay, great. And I believe you also elaborated on this a little bit in your last slide, but maybe talk a little bit more on why HHT is just in certain places or organs.
Yeah, again, I could speculate. We don't know for sure, but it is possible, again, going along the theory that angiogenesis is a big stimulating or a big factor involved in AVM formation. It's possible that areas in the body where there's higher rates of angiogenesis or maybe higher rates of infection or inflammation or injury or things that stimulate angiogenesis, it's possible that's why we may be seeing AVMs in these specific areas and not in other areas of the body. But a lot more research needs to be done for certain.
Okay. And I believe this is the last question, unless some more happen to come up in the chat. But why is it not possible for one person to get two mutations for different HHT genes, e.g. endoglin and ALK1? And if it is possible, would this produce a worsened form of HHT?
Yeah, certainly a good question. So, in my opinion, probably having mutations in both, like an endoglin or ALK1, would probably be incompatible with life. It would probably be, it would prevent the function of these cells so much that it wouldn't even allow for life to begin in the first place.